Dolly ships.
Exactly. The one on the right, screen's right is Dolly. The next one, first one is on left. You have seen this diagram. It's not related to biotechnology anyway. I mean, it's a biotechnology approach, but the diagram itself doesn't reflect complete biotechnology. The
Clone cell.
Yes, maybe. It's a developing embryo, right? So with multicellular, at multicellular level, right? This is important because one of the biotechnology approach, which is going to be, I mean, it's already there, but it's going to be in many ways, it's going to be used in stem cell technology. For that, we need embryonic cells, embryonic stem cells, which is highly useful in many technologies, right? Second, it's stuck. Right. Right. So recombinant DNA, gene cloning, and pharmaceutical production. Some of the most important and widely used technologies, right? Biotechnologies which are being used. So you know what a recombinant DNA technology is? We have spoken about this in our last session as well. Where recombinant DNA is involving combination of the DNA of two different kinds of organisms, mostly unrelated species, right? Maybe a bacteria genome into human, or genome rather, bacteria gene into human genome, or maybe bacteria gene into plant, so on, or viral gene into anyone else genome. So it's important because DNA recombinant or recombinant DNA technology is a path-breaking technology, which is being done in the lab, and this has been paved way to many revolutionized products in agriculture, medicine, pharmaceutical industry, in research, in all the fields. Recombinant DNA technology holds the key. And of course, the modern medicine is going to be based on the recombinant DNA technology, DNA editing, right? CRISPR, everything. Because if everything goes well, we may see a technology revolution based on recombinant DNA technology and gene editing technology in all the fields, right? And gene cloning, of course, we can clone any gene of interest. We can produce whatever we need. Pharmaceutical industry is one of the highly beneficial industry as far as the biotechnology is concerned. We have seen that in the COVID-19 pandemic through our eyes. Right? Pharmaceutical industry has been benefited by the state-of-the-art research done over years and years, and COVID-19 scenario gave the benefit to the pharmaceutical industry to use those technologies. And we have seen vaccines being produced within 6 months from the day of infection, or from what we heard from China, where whenever we have heard, the whole world heard about COVID-19 infection, the first vaccine was ready in 6 to 7 months for trial, which is huge. Because if that has been said, say 5-10 years back, that we will be having a technology based on biotech research, that we'll be able to create vaccines within a year, everyone would have laughed at that person who claimed that statement. But that has been a reality this year or last year, that we have made so many vaccines, candidate vaccines, within a very short time, right, with different efficacy, and of course some of them are highly efficient in fighting the disease. So that is again a path-breaking technology, where we have been able to produce vaccines based on mRNA. So we will be talking about that maybe tomorrow, right? So what makes this technologies. The basic thing which is needed for a DNA recombinant technology or gene cloning or whatever pharmaceutical products we are going to produce is the ability to incise the gene at a specific place, so that the precision of these cuts, we have to cut the DNA at specific place, and that cut should be very, very, very precise, right? That makes this technology even more exciting, right? Earlier we had technologies where we were able to change the DNA, but we were not being able to target very specific DNA, maybe very specific organ nowadays, to cut those DNA in those specific cells or cell lines. So that specificity is given by restriction enzymes or restriction digestion enzymes. That has been one of the primary discovery of restriction enzyme has given the scientists the ability to cut the DNA at very specific place. Maybe not to the cell line level using the standard restriction enzyme, at least they would be able to cut the DNA at the specific level, right? You would have studied about restriction enzymes. So these fragments which have been cut and incised or separated can be used in cloning or maybe recombined DNA as well. So restriction enzymes, you would have studied about this, so I'm just briefing about them. Restriction enzymes are the enzymes which have been able to cut DNA at very specific sequences. These are called, there are some restriction enzymes which are not specific. But these restriction enzymes where we are speaking about in the DNA recombinant technology or gene cloning are very specific enzymes. That means they can recognize a specific order of nucleotide. We call it a sequence. So recognition sequences can be recognized by these restriction enzymes, and those recognition sequences can be precisely incised at the specific place. So here we are giving an example in this figure, an enzyme called EcoR1 restriction endonuclease. So restriction endonuclease are the nucleases. That means nucleic acid cutting enzymes, so digesting enzymes. Restriction means they are restricted to specific place, so they can understand and identify specific sequence. So EcoR1, you know how to name these, so how the names are given for the restriction enzymes? You have studied, right? What is meant by EcoR1? Can somebody explain? Do you know? You haven't learned it. Have you learned it or not?
No, sir.
Okay, so it's simple. Eco means the organism, E. coli, right? The letter represents the organism's name. I mean, the letters means alphabets. Eco, right? Eco means E. coli. R is the strain, right? The capital letter comes after that is the strain of that particular organism. 1 is the number of enzymes. I mean, the order of enzyme we call. This is the first enzyme isolated from E. coli strain R. That's the meaning of EcoR1. If we say EcoR2, then we say that's the second restriction enzyme identified from EcoR strain. Right? That's how the names are being given. It's simple, right? So I just want to make sure. All right. So hence EcoR1 has this restriction digestion sequence GAATTC, right, in reverse order. We call it as palindromic sequence, right? Palindromic sequence, you know palindromes? Palindromes? Okay. Palindromes are the words which are being read from left to right and right to left, right? In English, there may be some words, dog or something like that, dog, something. In there are Tamils, you know, if you read the word from left to right and right to left, it will give the same word. I can say some words in Tamil, but English I have to search for them. Right. So similarly, restriction enzymes have this palindromic sequence recognition ability. So what they will do is, say for example, EcoR1 will cut between this G and A. So as you can see, it will recognize this sequence, but this will cut at this specific place between G and A. As you can see, it has cut the other strand of the DNA, opposite strand of the DNA at the same position. So you can see once this DNA fragment is cut down, you will be having two different segments. Similarly, another enzyme may not be able to detect this particular sequence, but it can be a different sequence. Say, for example, it can be a TATT, something like that, right? So similar sequences, sorry, not similar sequences, different sequences are being recognized by different restriction enzymes. So this is one important property because we have identified so many of restriction enzymes so that we can target multiple regions at the same time as well with higher specificity. So based on the number of recognition bases, say for example, here it is 6 bases. You know what is a basis, right? Nitrogen bases. We call them as 6 base cutters, 5 base cutters, 4 base cutters, etc. Right? So these are some of the restriction enzymes examples, right? All these things are made, I have taken it from internet, so all these things are made from one company, Biolabs, but there are so many other companies which are making. As you can see, this is ACI1. You can see the recognition side. This will CCGC. So this is a 4 base cutter. So this will cut between these two C's. APO1, this is CC. The slash indicates the cutting position, right? CGG, CG, CG, CC. So this is an 8 base cutter. So this will cut between these positions. So you can see there are so many enzymes and so many different cutting positions. So this is how they will be cutting, and we have seen that. So when these cut are not in the middle like this, but on the sides, either sides, we call that cut as sticky end cuts. That means once it is cut, we will have a segment of DNA hanging. Right? You can see if that is cut in the middle, say for example, if you cut in the middle, you will have segments, 2 segments like this, right? There will be no overhang. We call it as overhang. It will be straight. Hence, it will cut at the sides of each segment. You will have sticky end with a hanging thread of DNA. Right? So this cut is known as blunt cut. That means we don't have any hanging segments. Right? So this is another example. So once you cut down these segments, say for example, here we have cut the segments with EcoR1. So imagine you have 2 DNA. One is your host DNA, your plant DNA, and you want to insert the Bacillus thuringiensis specific gene, and you have Bacillus thuringiensis DNA. What you have to do is you have to cut this with EcoR1 at specific place, and the same enzyme can be used to cut the Bacillus thuringiensis piece as well. So both of these segments will give you the compatible sticky ends. That means Bacillus thuringiensis will have the same cut like your host plant DNA. So what we can do is we can bind them together, right? So you can add this piece here, this piece here, so that your host DNA will be having a foreign DNA which is matching the sequence of the host DNA at that specific place. So this process is called as mixing and ligation. So that you mix the DNA from both organisms, so different kinds of organisms, and you ligate them. Ligation means binding them, right? So this is how a recombinant DNA is produced, right? So you cut the DNA of your host organism and the donor organism with the same restriction enzyme, so that you can have compatible ends of the DNA to be joined together, right? Because otherwise it's not going to be joining, because you know DNA is having this specific binding pattern of Watson and Crick model, right? A will bind with T, G will bind with C. No other binding pattern is correct. There may be something incorrectly done, but they are not correct binding positions. So what will happen after you cut the host DNA and the donor DNA with the same restriction enzyme? They are compatible. They are made compatible for binding. So that when you add DNA ligase and give the correct environmental conditions, that means temperature, pH, and everything, then they will start binding with each other. So that you will be getting a host DNA now inserted with your foreign DNA piece. So this is called as DNA recombination, right? In lab, DNA recombination in general, inside the cell is also happening, but this is DNA recombination inside a lab. To do this process and to do gene cloning and to produce cloned organisms, we need a cloning vector. So you know vector. What is a vector? General, what is meant by a vector? Maybe in your
Carry
class.
something.
Sorry?
Carry something.
Exactly. Carrying something, right? So it's a vehicle. It's a vehicle to carry anything. So cloning vectors in biotechnology means they are the carriers of any possible DNA or pieces of DNA material into the host, right? So they will be having those pieces of DNA, because this is important in higher organisms, because these DNA, how we are going to insert into the higher organism is a question. We can cut the host DNA, can cut the foreign DNA, but in natural setup, how we are going to insert them? So how we are going to combine them? You can't just inject the DNA into the cell. So we need something, a carrier, which can carry this foreign DNA into the host DNA. So these vectors, cloning vectors, will do the job for us. So this is generally can be an organism, the whole organism, or maybe a piece of DNA as well. So what their job is, as I've told earlier, they will carry the inserted DNA from one organism to another organism, right? So in DNA will be taken from one organism, and that will be, say, DNA taken, and that will be inserted into the vector, right? And then this vector will transfer to the host organism. So this is a vehicle to take the piece of DNA which is to be inserted into the host cell or DNA. And generally, they should be very small, because they don't have any other job other than to transfer this piece of DNA into the host cell. So they should be very small. Generally, circular pieces of DNA from bacteria will be used. Why the circular DNA or plastids are used is that they will be reproducing very quickly, right? So that they can be easily inserted and easily produced in large numbers, because we are not going to insert a one piece of DNA. We have to understand that as well, right? All these recombinations and cutting has a probability, right? You don't expect restriction enzyme. Say, for example, you add restriction enzyme to a DNA. You don't expect 100% of the cut is going to be happening. You expect certain probability, say 70%. So we need multiple numbers of copies of these clone vector, so that 70% of them will be going and binding with the specific DNA. So we need more copies. So we need a DNA piece or a clone vector which is capable of multiplying quickly and producing large number of DNA pieces, circular DNA at a very short time. So that is also a selective advantage of clone vectors. And bacterial plasmids are the number one choice. Plasmids are circular DNA, which is normally found inside the bacteria. The most important advantage is bacteria can be readily transmitting plasmids. You can transfer plasmids into bacteria very easily. And of course, they will transfer those plasmids within a colony. So if you culture a bacteria inside a colony without reproduction or even producing any crossovers, the plasmids can be transferred from one bacteria to another bacteria within the medium, right? It's like popping out of the organism and taken out by another organism. That is a normal process of plasmid transfer between bacteria inside a colony. So they are very flexible pieces of molecules, right? So that they can be used. And other than the bacterial plasmids, viruses can also be used. And yeast also can be used, right? And they have their own limitations. We will see some of them. And plasmids, as I've told earlier, circular pieces of DNA. You would have studied about them, right? So it's found in the normal bacteria cells, right? So they have an independent replication cycle not associated with normal bacterial chromosome. Even without replication of bacteria, I mean the cellular replication, plasmids themselves can replicate readily throughout the lifetime of a bacteria. So these plasmids are commonly constructed vectors. Most of the time in research and all these things, we use plasmids because they are hassle-free. Sorry, unlike other organisms. So, but other vectors are also being used. Specifically in animal biotechnology, we prefer more readily insertable vectors, not as plasmids, because mammalian cell line and animal cell lines are different from plant cell lines, right? They don't, they have their own immunity against these kinds of. So we need a vector which can target specific organs, specific places of animal cell lines. So we prefer other vectors over plasmids. So there are viral gene transduction agents available. Transduction means from what you insert something into a cell through an agent. So there are viral gene transduction agents available. So many of them are available. They have their own strengths and weaknesses, right? They depends on the type, depends on the need. They may have their own goods and bad things, not good and bad, pros and cons, right? Good strengths and weaknesses within themselves. So most commonly used viral vectors are retroviral vectors. A retroviral vector means, what is retrovirus? What is a retrovirus?
The protein kill.
Sorry? What is a retrovirus? You have said something, I couldn't get you.
Which contains RNA is the genetic material.
Exactly. So they are RNA virus. That means they are major genetic material found inside the protein capsids or protein core is RNA, right? Most of the animal virus are retroviral, but not always, most of them. So they are potentially very useful for gene transfer in dividing cells. That is important. because gene transfer needs to happen inside a cell line which is readily dividing. So retrovirus are very useful in gene transfer into dividing cells. But for stagnant cell, right, what we have to do is we may need to add additional things into the retroviral particles. We call this process a retroviral packaging, right? But the problem is, yes, as I've told earlier, they are inability to infect in resting cells. And there is potential side effects as well. Hence, the animal virus, they tend to alter the animal genome by themselves without our desirable alteration. So what we can do is, again, retroviral packaging might help. So this is a retroviral packaging method which has been suggested. This is not the only retroviral packaging available. What we do is we target this retrovirus here, and we will be having another vector set which is try to envelop this retroviral vector, so that the retroviral vector will carry the introduced gene into the cell, but at the same time envelope vector set will prevent it from causing any harm, as well as it will allow additionally push the retroviral vector into resting cells as well, right? So you have to combine these two different cell lines, not cell lines, vectors into a plate, and you have to culture them, right? And after 48 hours, you can harvest them. And now the retroviral vector, which is packaged, that's why we call it as package. We pack them with the other envelope vector set, right? So that they will be readily transfused into the host cells. There are many new agents are also being proposed, right? The other thing is adeno-associated virus, which is not a retrovirus, but a DNA virus. So this has also been identified to infect both resting as well as dividing cells. So this is an additional advantage of using this kind of virus. So generally, a potential viral vector agent should be able to concentrate it in high titers, right? So that means they can be able to be having pooled into large quantity in a specific volume, right? So that they can be treated to large organs or larger cell pools, right, inside the body or inside the animal, not in vitro, in vivo, where we insert them into the animal, right? So, and also they should be able to have a maximum capability of infecting different kinds of cells, right? So that we can target various cell lines and various organs. And they should be having flexibility in the application because of this. And they should be having very low immune response from the host line. Because when we insert the viral vector into the animal, right, not inside a cell line or a tissue cultured in the lab, but into the real animal, there will always be immunological response, right? That means there will be always a response from the host animal to block the entry of this virus. So the good viral vector should have a relatively low immunological response expressed by the host, so that they are relatively non-immunogenic. So they could be made replication deficient, right? So that they can be packed with ease as well, right? So they should be easily handled and they should be easily packed so that the transduction is more effective with less side effects as well, right? So we'll go into clone a gene. So we will be using, you would have studied about this, so I just put the process here. So what we will do is we will use the same restriction enzyme to cut both the gene and cloning vector. It can be plasmid or it can be viral vector. So we have to mix them in a tube, and DNA ligase will be added to bind them. And then these plasmid bacteria, now a recombinant bacteria, plasmid, will be cultured, right? So there are various selection procedures to grow, depends on the nutrient or depends on the antibiotic. Here we hear what they have said is about antibiotic, but it can be based on the nutrient as well, right? So that you can culture the recombined bacteria into a cell. So this is the procedure given in a nutshell in a single diagram, right? So you can see the plasmid is cut by the same enzyme, the gene itself cut by the same enzyme, so that the compatible ends are produced for both of them to bind. So once the DNA ligase is added, everything will be bound together. Bound, as you can see, there may be mismatches as well. As I've told earlier, it's not 100% efficient procedure. There is a probability of efficiency as well. So there may be mismatches, but only the correctly matched cloned plasmid or bacteria will be developed further. Depends on the selection by culture media. So these are some of the applications. Quantities of proteins, right? You have said about the insulin. It's easy to handle them, right? And you can produce large number of proteins in prokaryotes, because prokaryotes are very simple organisms with a very simple gene setup or genome, we can call it. And so that they can be easily manipulated. So that the gene transcription and translation is very simple. Once you know the exact places which is required for the transcription and translation, you can manipulate them. But doing that in a eukaryotic cell is much more difficult, because they possess a thing called introns. You know what is an intron? Introns are the non-coding regions of DNA. They are not the only non-coding regions, but they are making a very large amount of non-coding regions in a specific genome in eukaryote. So that it is highly, not highly, it is very difficult for the eukaryotes to be used in this cloning process. What we can do is we can transfer the DNA into mRNA, reverse transcription process. Sorry, not to transcription. And then what we have to do is we have to reverse transcript this mRNA into DNA. Then we can use that cDNA, or we can clone DNA or complementary DNA to use in the cloning process. But that is an additional step we should be taking when we use the eukaryotes. So this is the process of producing insulin inside a human cell, right? So what we have to do is we have to isolate human cells and grow them, and then we have to isolate DNA, and then that has to be cut, and plasmid has to be made, and then we have to insert the insulin gene, the complete gene with all the required regions to be used in the transcription and translation process to be inserted. So once they are inserted, and the new recombined bacteria can be cultured, right, into insulin producing bacteria. So it's a very looks a simple process, right? You can see the reactor, right, the culture system where you can use these things. And this is the insulin. We call it as this is one brand, Humulin, right, which is produced by a company. So this is how it has been done, right? It can be the similar for most of the products. It's not to be insulin, it can be any pharmaceutical product which has been produced by DNA recombinant technology. It's the same, right? So I will show you some of the pictures where we use them in the animal husbandry industry. This is genetically engineered salmon, right? As you can see here, This is the genetically engineered salmon. Both of them are 14-month-old. These are non-genetically modified standard salmons. As you can see, this is huge, right? Because they are supplemented with additional growth factors to biotech process or genetic engineering. This is Dolly. We have seen that in the first picture as well. The first cloned, completely cloned whole organism, right? Whole animal, not organism. These are again cloned pigs, right? And there are various needs for cloning animals, right? One thing is if you can find a breed which is very rare and it has superior quality in the husbandry, animal husbandry or agriculture industry, then you can improve the herds. Or maybe you can clone the animals for pharmaceutical production, right? Testing and all these things, testing in animals, we have ethical issues, right? Because wild animals are being used, even lab-bred animals are being used. But the problem is you don't know the exact genetics of that particular organism. Because nowadays, hence as I've told earlier, medicine is more target oriented, right? Very specific medicine are going to be given to each people, depends on their genetic makeup. So if we have a facility, technology to produce animals with known genetic makeup, then it is going to be huge, right, in the pharmaceutical industry as well, because we can test those things in these animals with known genetic makeup, right? Or sometimes it is aesthetics, right? Cloning an animal or a plant or anything. This is the first cloned horse, right? Mother and the cub. The first cloned mule, right? So mule has also been cloned. And interestingly, Chinese or Japanese people have cloned chimpanzees. That's the highest level that has been officially cloned. But not cloning, but the Chinese doctor has been arrested to produce embryos, human babies with modified DNA, right? Last year, 2020, if I'm correct, or 19. Because what he has done is he has used CRISPR technology to edit the embryo, right? So that a specific genome has been produced in those embryos. So that is also possible. Human cloning is also possible. So this is the cloning process. I think you have studied the cloning process. I have. Right. So this is an interesting aspect. We have talked about cloning whole organisms, but cloning tissues and organs is also important. Right. Medical field is looking for this technology because there are so many organ replacement. Kidney is one of the biggest problem. Kidney replacement is one of the biggest problem, or liver replacement is one of the biggest problem in medical industry, because you have to find a matching thing, right? Kidney or liver, whatever it is. So if we can culture kidney inside a lab, right, with specific genetic makeup, with specific, right, just like ordering your dress, right, tailor-made organs, then that is going to be huge in medical industry as well. And culturing tissues, and one interesting, I will introduce that in my next session. Culturing tissues is another important thing, right? If you can culture your meat inside the lab, and that is going to be huge as well. You can culture your own, not own, whatever meat, beef or mutton, you can tissue culture them, right? Based on your desired DNA, whatever you don't want, right? Maybe with less fat or maybe with added ingredients. That's also important. Right. Stop this here. Right. So what we are going to learn next is it's about probes, DNA probes. I will give an introduction and then we will talk about the molecular vaccines. Right. Tomorrow, 9 o'clock. Okay.